really happy to be hosting today Dr. Daniel Foulkes to discuss his recent PLOS ONE Lab Protocol paper entitled Stable Topical Application of Antimicrobials Used in Plumbing Rings in an Ex Vivo Porcine Corneal Infection Model and the associated detailed protocol in protocols.io. Daniel's research career began in 2013 at the French National Center for Scientific Research and University of Paris-Saclay under the mentorship of Dr. Ruxandra Graf. As a research assistant, Daniel adapted Prussian blue staining to visualize iron-based nanoparticle uptake in Staphylococcus aureus, infected macrophages, and applied analytical chemistry techniques to quantify antibiotic nanoparticle loading, an experience that sparked his interest in bacterial pathogenesis and host-pathogen interactions. Motivated by his passion for molecular biochemistry, Daniel pursued a PhD with Professor Patrick Ayres at the University of Liverpool, where he studied TRP2, a cancer-associated pseudokinase. Daniel developed expertise in recombinant protein expression, assay development, high-throughput screening, and cell signaling, ultimately discovering the first small molecule modulator of TRP2 and contributing to our understanding of its role in acute myeloid leukemia. In 2018, Daniel joined an interdisciplinary team at Liverpool, bridging the Department of Eye and Vision Sciences and Biochemistry, working closely with Professor Kay and Ferning and collaborating with Dr. Tam at the Cleveland Clinic in Ohio, United States. Daniel established a discovery pipeline to identify inhibitors of Pseudomonas aeruginosa, XOU, a potent phospholipase linked to several corneal infections. Through collaborative in vitro and cellular screening efforts, Daniel's team identified promising XOU inhibitors. With Dr. Tam, Daniel explored the therapeutic potential of XOU inhibitor drug leads in a mouse corneal infection model. Now, 6 years of this project, Daniel continues as a researcher co-investigator at Liverpool, driving efforts to characterize the mechanisms of action of XOU inhibitors and their pharmacokinetics. To improve the translational relevance of ocular infection models, Daniel developed an innovative ex vivo porcine corneal infection platform using medical adhesive and plumbing rings. This method allows precise and stable topical drug delivery and facilitates more physiologically relevant studies of host pathogen interactions and antimicrobial efficacy, highlighted in the recent PLOS ONE publication that we will discuss today. Dan, thank you so much for talking to me about your paper and your protocol.
Oh, you're welcome. Thank you for the very nice introduction.
So to kick off, can you briefly explain what the protocol is about, how to use it, and why it is relevant?
Yes, of course, the protocol describes a very simple but effective method for studying microbial keratitis in an ex vivo porcine cornea. And we use plumbing rings and medical adhesive as an innovative approach to create a seal or a reservoir on the corneal surface where we can localize bacterial infections and precisely apply and monitor topical treatments. And this allows for a controlled and reproducible assessment of their efficacy and to study host pathogen interactions. It's a relevant model for eye research, bridging the gap between traditional in vitro systems and more complex in vivo systems, but offering a cost-effective and ethical approach for testing novel therapies.
So to further understand the significance of your protocol for the field, So can you elaborate on the motivations to optimize the protocol? And as an elaboration of that, which protocols were already available when you decided to engage in this project, but those protocols didn't work for you?
Yes, it's a widely adopted model, and it's a very important tool. But previous protocols often relied on simply pipetting the pathogens onto all the drugs onto the corneal surface, and which by aggregate led to significant runoff into the well plate, and so not on the cornea. And this was really difficult in experimental scenarios to make sure we had consistent infection and treatment delivery, where we'd have some eyes that were infected to different degrees or not even at all. Recently, well, a few years ago at the University of Sheffield, they had a fantastic approach, a very innovative approach, where they used custom-made glass molds, and these were very effective at keeping infection in one place and topical treatments there. But glass molds weren't readily available to us, and they required access to specialized glass blowing facilities. So our motivation stems from a way to make this cost-effective, simple, and accessible to researchers, and so people can use them without implementing specialist equipment.
So because your protocol is more affordable and its adoption could be widespread, because if I understand, we're talking about just
Right.
plumbing rings, right? That can be purchased in any hardware store,
Exactly, yes.
potentially. So what kind of experiments or applications will this protocol enable that weren't possible before?
So, yeah, as you said, the plumbing ring reservoir maintains both the pathogen and the treatments exactly where you want them to be, central to the cornea in the experiment. It improves the consistency of the experiments, and then it allows more accurate interpretation of the outcomes. And moreover, because the materials, yeah, they're cheap, easy to apply, the protocol kind of democratizes access to ex vivo microbial keratitis models, and especially for labs without high-end infrastructure. But the key advantage is, compared to previous protocols, is the spatial control that you have.
So I understand that before developing the protocol, you were inspired by the innovative method developed at the University of Sheffield.
Yes.
How did you come up with the idea of using plumbing rings? And whilst you were developing the protocol, is there anything else that you tried that failed and didn't make it to the final version of the protocol?
Yeah. So we settled on plumbing rings and we thought that it would work. But there were several iterations before that. We tried vacuum grease and Vaseline to try and make an even seal where we could apply stuff to the top, but there were mixed results, and more often than not, you would just spring a leak in about 80% of your samples. But then my supervisor, Stephen Kay, he's a clinical ophthalmologist, and he said we should try some medical adhesive. So I popped round to the hospital. Fortunately, I didn't have to wait in the clinic because the waiting times are atrocious at the moment. He took me to one of the consultation rooms, gave me some medical adhesive, and said, try this. And hey presto, it worked. And it worked really well. Damaging the epithelium didn't seem to be a problem, and histological analysis showed that the using the medical adhesive didn't have any real impact on the surface epithelium. It was quite a gentle approach, but effective, and allowed us to have an even seal.
Your protocol on the platform and the paper are very nicely written. You have supplemented the protocol with pictures to allow people to really understand how the procedure should be done. But to provide the users a better insight, what are the most critical steps? What parts of the protocol are particularly tricky and others might make a mistake on?
So it's the actual positioning of the plumbing ring and making sure the plumbing ring is the correct size as well. We advocate the 5mm ones, which worked well for us across the kinds of eyes that we were getting from the abattoir. You can use smaller ones if you're studying different types of infection, like HPV. But if you need to make sure it's firmly attached, making sure there's no leakage. We recommend allowing some drying time as well for the adhesive and visually confirming as well. I think that needs to come across quite well, because if I've tried to add topical treatments too early, there can be some leaks. But by the time I've dissected and sealed 10 eyes, I come back to the first one, and then it's okay. We also describe how to debride as well, and we suggest that you can use a needle or a scalpel to scrape the corneal epithelium, but personally what I found was if you were doing it with just a needle or a scalpel, you would be getting mixed results, especially small micro changes in how you move your arm or use strength can impact this. So we advocate, and this was adapted from a previous protocol again before the Sheffield one I discussed, where her name was Stephanie. I forget her second name, unfortunately. Stephanie used 70% ethanol to loosen the epithelium, followed by gentle debridement, rather than just purely mechanical debridement. And so that was, it might not be as physiologically relevant, but we found it was more consistent. And so I'd advocate for that one, but then again, it depends on what you're comfortable with. You could do either.
So without being an expert myself, and obviously without me trying the protocol, from the description, it seems artisanal. And
Right.
but on the other hand, it enables people to do screens of, let's say, antibiotics against bacterial infections of the cornea. Can you imagine how this protocol could be further optimized to make it high throughput for small molecule screening?
So not in the classical sense. So the nature of the protocol, like you said, very artisan, lots of handling and preparation with whole intact pig corneas. So that would make a true high throughput adaptation challenging. At present, I'd say a single researcher could manage around 36 eyes per experiment if you're using multiple 6-well or 12-well plates. But beyond that, it's the main caveat, the main bottleneck is the dissection and the preparation steps. So if in theory there could be an automated dissection tool, then potentially yes. But such tools would need to be specifically designed and developed. There are parallels with the precision cut lung slice model, where researchers have used like whole lung slices and vibratomes to actually cut out small sections and use biopsy punctures to allow high throughput sampling of thin tissue sections. But this wouldn't really preserve the integrity of the cornea, which is actually central to what we were trying to look at. You could look at sections of epithelium potentially that way in a high throughput way. So actually, yeah, there is development potential there. But the main strength of our protocol is maintaining the anatomical fidelity of the eye. And so that researchers can more accurately mimic drug administration, study pharmacokinetics, infection in the whole eye.
Daniel, I would like us to change gears a little bit now and talk about the motivations for you to send the protocol to PLOS ONE to be peer-reviewed as a standalone methodological paper.
Sure. Well, the peer review process ensures that the work is validated and it's reproducible, but also citable as well. The protocol solves a practical problem that I found that many researchers do encounter. And as scientists, we can't do it alone. And we're also committed to open science. And actually, thanks to PLOS ONE and Protocols.io, we've already gained significant visibility. A lecturer from Cardiff University recently reached out to me, and he was interested in employing this model in his own research. And so, in fact, he's going to visit Liverpool for a few days in a couple of weeks, and I'm going to teach him how to run the assay.
That is exciting. And I would like to get to that point at the end of the conversation. But going back to the peer review, can you tell us how and if the feedback that you receive from the peer reviewers shape the final version of your protocol?
Yes, so we had originally mentioned in the discussion that from the histological analysis, it would be possible to quantify intact epithelium and stromal edema. And the reviewer's response was, okay, do it. And so, yeah, we did it. And in doing so, it gave us an additional objective metric for measuring infection severity in our model, which ultimately strengthened the paper. And to our knowledge, this kind of quantitative analysis of stromal edema, which we did in ImageJ and monitored stromal space, it hasn't been widely applied in similar ex vivo models. So it might represent a useful precedent for future studies.
Why did you decide to make the peer review history public together with your paper?
So because we're inviting the community to see the evolution of the protocol, it encourages trust in the work. It also helps others understand the considerations that shaped our rationale and our reasoning and our approach. And so in the future, there's an understanding of the protocol's rationale when we publish research that uses the model, which we intend to do soon.
So, and my final question, you briefly touch on this point of how the publishing of the paper and the protocol has prompted collaborations. The paper is very young. It was just published a month ago. So my question might be speculative at this point, but what do you think has been or will be the significance of publishing this paper, this protocol for your career?
Yeah, so the paper is young, but I've seen it's already gathered some serious interest and quite a number of reads already, which is really encouraging. And the protocol itself is becoming a central platform in the lab for studying host pathogen interactions. And we aim for it to be employed by colleagues that are studying infection dynamics, even if their research field is not centered around the eye. It's opened up collaboration, like you mentioned. Imran from Cardiff will be coming to Liverpool soon, and he seems like a great guy, and I look forward to collaboration in the future. The work itself has helped shape a larger translational research pipeline, which is leading me to be able to pursue new grant opportunities.
Dan, I don't have any other questions. I don't know if that you would like to mention anything that hasn't been discussed so far.
No, just thank you so much for having me. It's been an absolute pleasure. And just it really highlights how simple innovations, even just plumbing rings and medical adhesive, can form practical problem solving towards advancing understanding and making small but really useful improvements to established models.
Thank you, Dan.
Thank you.